Hello, future doctors. Welcome back to GCSE Biology by Best Medicine. This particular series, again, is concerned with AQA syllabus, and this is going to be the final lecture for Chapter 2, Organization. In this particular lecture, we'll be talking about the respiratory system, and we'll be talking about organization in plants. Previously, we have finished the digestive system and the cardiovascular system, the circulatory system. So what is the respiratory system? The respiratory system is the system of the body that is involved in gas exchange. And we have already looked at, when we were doing the gas exchange of materials chapter, a little bit of how the alveoli are adapted for gas exchange. So the respiratory system, which will include the alveoli, helps in oxygenating the deoxygenated blood. So what is the anatomy of the airway? Well, for this, we'll have to, I think, draw a diagram here. So we have a person, and this person will have a nose, right, through which the person can breathe. And the person can also breathe through the mouth, right, through the mouth. So as the person breathes, right, air is going to go into two things. Number one, so these are called the nostrils, through which air is going to go into the nasal cavity, and air is going to go into the mouth, the oral cavity. And then eventually it's going to go into the pharynx. Pharynx is just a fancy word for space behind the mouth and the nose. Now from the pharynx, it enters the throat. The throat is a special place because it's got this division of the trachea in the front and esophagus at the back. So at the back, we've got the esophagus. That's the food pipe. We don't want air to go into this. We want the air to go into the trachea. So air is going to go into the trachea. On top of the trachea, we have a tissue here that blocks the trachea to prevent any food from entering. And this particular tissue is called an epiglottis. Okay? Epiglottis. Now, just below the epiglottis, in the trachea, we have the voice box, also called the larynx. Larynx is the voice box through which we're able to, so air, when it leaves, passes through the voice box, making it vibrate, and we're able to hear sound. Right now, as I'm speaking to you, my larynx is vibrating because air is going through it. So that is the voice box. Now, air will enter the trachea and go down. So here's the trachea. It goes down and then eventually it bifurcates into two bronchi. Okay, singular bronchus. So all this is the trachea as air is coming down on this, and then it bifurcates into two bronchi, left and right main bronchi. And each of them will enter into lungs. So here's a lung, for example. Right, and then here's another lung on the left side. These bronchi, they divide into further smaller bronchi, and then eventually these divide into smaller portions called bronchioles. And then the terminal portion of bronchioles contain the alveoli, okay? These balloon-like sacs, which will be called alveoli. So again, we start from the nostrils or the oral cavity, the mouth. Air is going to go into the nasal and oral cavity, from where it goes into the pharynx, and from the pharynx in the throat, it passes under the epiglottis into the trachea through the larynx. The trachea is right here. So that is the trachea. And through the trachea, air goes into the bronchi, right? Singular is bronchus. And then from the bronchi, it's going to go into the bronchioles, and then eventually into the alveoli. So that is the anatomy of the airway. Now, how are different portions adapted for different for airway functions? Well, first of all, we've got the nose, the nasal cavity, right? That's one of the most fundamental units of being able to breathe. Now, the nasal cavity has a couple of things going on here. Nasal cavity is able to produce mucus here, right? So it's producing this mucus through which air is passing, and this mucus, what it does is that it is able to hydrate the air, right? It's going to make the air moist so that the air that we breathe it doesn't turn our lung into a dry sponge, right? It's a very, we want the air to be soft and moist. So this is mucus that is being produced by cells here called goblet cells. I'll draw them as well in a bit. And these are cells called goblet cells, which are present in the nasal cavity, and they're producing mucus. So mucus, again, is a watery substance. We talked about mucus when we were doing the saliva as well. And this softens hydrates the air, right? Makes the air moist. Secondly, this mucus, because it's sticky, it is able to trap any dust particles and any flying bacteria. The other function of the nasal cavity is that it's got sensory receptors here that are able to sense smell. So at the roof of this of the nasal cavity, it's got these sensory receptors which are able to detect the sensation of smell. So sensory smell receptors. And whenever you smell something, what happens here is that these receptors, they give off electrical impulses which travel through a nerve to the brain. Okay, so each of these is connected to a nerve, and then the nerve goes and takes the sensory smell information to the brain. So that's the nasal cavity. Then we've got the pharynx, and then eventually through the larynx we enter into the trachea. Trachea's adaptations are also important for AQA, so we have to understand the structure of trachea and how it is adapted for its function. But before we dive deep into this, let's just have a look at what the trachea should have, right? What the trachea should have to be able to support this entry of air, smooth entry of air into the lungs. Well, one thing that we can sort of assume is that the trachea should at least always be open, right? It shouldn't be blocked. It should always be open so that air has an unobstructed flow to the lungs and then away from the lungs. The other thing that the trachea should have is maybe a lining of mucus, just like we have in the nose, so that it is able to trap any dust particles and any bacteria and also soften or moisten the air. And the third adaptation that the trachea should have, right, just by looking at it, is that if you've got mucus, then you want to remove that mucus as well, right? You don't want the mucus to start building up in the trachea, so you want to have something to remove the mucus as well. So let's see if the trachea is adapted for these functions or not. So what we're going to do is we're going to take a portion of trachea, right, and zoom into it on the right side. Okay, so here's the trachea. The trachea has these C-shaped rings of cartilage, okay? These C-shaped rings of cartilage. What is cartilage? Cartilage is a strong, rigid connective tissue that is able to hold body structures rigidly. So for example, at the ear, right? The ear is a cartilage. Your nose, the front of your nose is a cartilage. It's a little bit soft, but it's still rigid and holds the structures like the ear and the nose in shape. You also have these in the trachea. So these are C-shaped rings of cartilage. And what they do is that they keep the trachea open all the time, okay? Make sure that the air flow is unobstructed and smooth. So C-shaped rings of cartilage. Why are they C-shaped? Why are they not completely surrounding the trachea? The reason for this is because they've got some muscles at the back, okay? And these are smooth muscles at the back. That's why they're not completely surrounding the trachea. So C-shaped rings of cartilage make sure that the flow of the air is unobstructed. The other adaptation for the inside the trachea is that you've got these cells that produce mucus. So the epithelium of the trachea, the respiratory epithelium, is quite unique, okay? So it's got these two types of cells. One of them, this big one here, right, and maybe another big one here, are the goblet cells. We call them goblet cells, okay? Goblet cells. And goblet cells' only function is to be able to secrete mucus. So what they're essentially doing is just secreting mucus. And remember, mucus is this watery substance, right, that hydrates the air and traps any dust particles. So that is the mucus that is being secreted by goblet cells throughout the walls of the trachea. Now you want to remove this mucus as well. So to be able to remove the mucus, the normal cells, the other cells of the respiratory epithelium, the respiratory epithelial cells, they have these hair-like projections in front of them, okay? They have these hair-like projections in front of them that are able to block, or they're able to circulate the mucus. So for example, over here, these hair-like projections, and what they do is they move around and they push the mucus upwards. These hair-like projections are called cilia. The cilia, they help circulate mucus. And again, what do the goblet cells do? They produce mucus. Which, number one, hydrates air because you don't want dry air to go into your lungs. And number two, traps any dust particles and bacteria. Okay. Now, as this mucus is circulated upwards, right? This mucus is circulated upwards by the action of the cilia. Eventually, it reaches the throat. So again, let's go back to our diagram. So here's the trachea, right? And then we've got the mucus that is being circulated upwards in the trachea. When this mucus reaches the throat, it is taken into the esophagus and swallowed, so that all the mucus eventually goes into the stomach, and any bacteria, any dust particles are broken down or digested. Okay? And that is a summary of the adaptations of the trachea. All right, so let's have a look at your need notes. So here is again a diagram showing the anatomy of the respiratory tract. The upper respiratory tract includes the nasal cavity, the mouth, the pharynx, and then the lower respiratory tract includes the larynx, which is the voice box, the trachea, as you can see here, with its rings of cartilage, right? And then eventually it divides into these bronchi, and then bronchi further divide into bronchioles, and then those bronchioles would eventually give you alveoli. Okay? Now, something very important that is mentioned here in this particular image is a muscle that is present right below the lungs. This muscle here is called the diaphragm. Okay, and this diaphragm is going to help primarily, it is a diaphragm that helps us breathe in and out. So we'll come back to it in a bit, but let's have a look at the trachea and bronchi's need notes. So again, trachea and bronchi, they have rings of cartilage that keep the airway open. They have goblet cells that secrete the mucus, and mucus helps trap any dust particles and humidify the air. And they have respiratory epithelium, which essentially means the ciliated cells that we saw, and those ciliated cells help circulate the mucus, remove the mucus up to the throat where it will be swallowed, okay? Now, a quick disease tie-in, I want to mention this here. Although it is repeated later, this disease, you're supposed to be aware of this in AQA GCSE. This is called cystic fibrosis. Okay, cystic fibrosis. And that's a genetic disease that belongs to a category of something called autosomal recessive diseases. Right now, you would not know what recessive is. You might know what an autosome is previously from our studies of chromosomes, but autosomal recessive disease. So cystic fibrosis is an autosomal recessive disease. What happens in cystic fibrosis is that you have this mucus that is being secreted by the trachea, the epithelium, the goblet cells. The mucus becomes very, very thick. Okay, the mucus becomes very, very thick and sticky, so that it is very difficult to remove it. When the mucus is very difficult to remove, then all the bacteria is going to pile up here, right? And it's going to cause a lot of coughing. So it's going to dilate all the bronchi and bronchioles. So it's going to cause a lot of problems there, infections. And that is cystic fibrosis. Sometimes they give you questions about cystic fibrosis related to it being autosomal recessive. So you would not directly be asked what happens in cystic fibrosis. However, you need to know that it is an autosomal recessive disease, and this will be mentioned in the inheritance chapter. So the mechanics of breathing. This is a low yield topic. This is not a very high yield part of AQA GCSE, but basically the way breathing works again is the diaphragm. The diaphragm is a muscle that is present right below the lungs. When the diaphragm contracts, right, it flattens down. when the diaphragm contracts, you have to remember this, okay? Generally, you might sort of through normal intuition, when you look at it, you might think that, okay, if the diaphragm contracts, it probably is going to arch upwards and push the lungs. No, when it contracts, it actually flattens down, okay? So when you're putting in the work and putting in the energy in the diaphragm muscle, it's going to flatten down. And when it flattens down, it increases the volume here. And then increases volume here, it decreases pressure inside the chest cavity, inside the thoracic cavity. So when the pressure inside the chest decreases while the pressure outside in the atmosphere is greater, then air rushes in to the lungs, okay? At the same time, when air is rushing in, the rib cage is going to arch upwards. The chest is going to expand. Similarly, when you want to breathe out, then the diaphragm is going to relax. When it relaxes, it actually arches upwards, as you can see here. So when it arches upwards, it's going to push the lungs and squeeze the air out. It's going to increase the pressure here, and the pressure outside will be comparatively lower, so air is forced out of the lungs. And that is breathing in and breathing out. This is further assisted by muscles that are present in between the ribs. You see the ribs of the rib cage? In between those ribs, you have these muscles here. So in between the ribs, let me just draw this in green. Muscles are not green, but so between the ribs are muscles. Ribs are also called costal bones, okay? Costal bones. And so the muscles between them are called intercostal muscles. Intercostal muscles also help out in breathing in and breathing out. So again, breathing in. Breathing in is also known as inhalation or inspiration, and it is the process of taking air into the lungs. Now what happens is that the diaphragm contracts and it flattens down, right? The diaphragm moves downwards and the rib cage expands, increasing the volume of the thoracic cavity. This decreases the pressure inside the lungs, causing air to rush in. Oxygen is then absorbed into the bloodstream through the alveoli in the lungs. What about breathing out? Breathing out is going to be the opposite, right? The diaphragm is going to relax. When the diaphragm relaxes, then it's going to arch upwards. The rib cage will come down, right? And this will decrease the volume of chest cavity, thoracic cavity. So the increase in pressure inside the lungs causes air to be forced out of the lungs and expelled from the body. So remember, during exhalation, the reason why we're doing exhalation is so that we can remove the waste products, carbon dioxide, and get the fresh air in. So CO2, a waste product of cellular respiration, is removed from the bloodstream and expelled from the body through the respiratory system. These processes are essential for the exchange of gases between the body and the environment, facilitating cellular respiration and maintaining homeostasis. Okay, and of course this is an automatic response, right? We breathe automatically. Until we consciously try to become aware of it, normally we just breathe automatically, which I have been doing throughout this lecture, right? So we have a meme on it. My lungs breathe automatically, me thinking about it. And my lungs, well, now I'm not doing it, right? As soon as we start thinking about it, it becomes conscious, and then it stops being automatic breathing. So how does the gas exchange occur in the alveoli? I'm just going to go over this very quickly because we've already done this in the gas exchange chapter. So gas exchange, alveoli in lungs. Alveoli are sac-like structures in the terminal portion of lungs which allow gas exchange to occur. So what you're getting is that you're getting this air, inhaled air, which contains oxygen, to the alveoli, right? So what will the alveoli have? Well, here's the alveolus again, right? And here's a blood vessel that is coming in, and it's bringing in fresh deoxygenated blood, right? This deoxygenated blood is going to have increased CO2 concentrations and low oxygen concentrations, while the air in the alveoli that is freshly coming in, this will have increased oxygen concentration. Remember, oxygen is like 20% in the air, while CO2 is 0.095%, so very low CO2. So this generates a diffusion gradient to be set up for oxygen to move into the blood and CO2 to move outside of the blood, resulting in the blood becoming oxygenated. So now the blood is oxygenated. It's got more oxygen, less CO2, and eventually returns. Now, the way the alveoli are adapted for this is that, number one, they have a very thin walls here. So alveolar walls are very thin, right? Very, very thin, one cell thick. And also the capillary endothelium, the epithelium of the capillary, is also one cell thick. So both the epithelium of the alveoli and the capillary are one cell thick, which means there's a short diffusion distance, allowing faster diffusion. The second adaptation is that you have fresh blood coming in and already oxidized blood leaving, which maintains the concentration gradient. Okay. The third adaptation is that you have a very large surface area as compared to volume ratio. These are very, very flat cells, which allows increased surface area to volume ratio for faster diffusion. And lastly, we have a thin layer of moisture here that allows the diffusion of gases to dissolving of gases so that they can go across. Okay? And these features are mentioned in your notes. So we get a meme. Maybe I should smoke for the first time. I mean, what could go wrong? And then my lungs. My word. And yeah, that's pretty much it for the respiratory system. So pretty brief for AQA GCSEs. And next up, we have organization in plants. I think most of the organization in plants, you guys are already familiar with most of it. Okay, so plants are also made up of tissues and organs, and we need to understand the tissues and organs of plants as well, right? So some important tissues in plants include, we're going to do number 1 and number 6 first. So number 1 is epidermal tissue. Epidermal tissue, again, just like the epithelial tissue in humans, epidermal tissue is, it surrounds essentially the outer and inner layers of organs. For example, the upper epidermis and lower epidermis of the leaf. And we'll have a look at it in a bit. So in leaves, epidermis is transparent, allows light to pass through into the photosynthetic cells. We're going to have a look at this in a bit. Then number 6, meristematic tissue. I think you should be well aware of this by now because when we were doing cloning of plants, we talked about this. And also differentiation of plants, we talked about this. Developing region of the plant contains stem cells that are able to form any kinds of cells in the plant. And we can use them for cloning using meristematic stem cell tissue culture. Okay, so developing region of the plant contains meristematic stem cells. 2, 3, 4, and 5 we're going to do after we finish the leaf diagram. So again, how does a leaf look like? Well, here's a leaf, right? The leaf on a cross section, you've probably seen a leaf before, right? So a leaf kind of looks like this, right? And it's got these veins, and then these veins essentially bring in the water and the nutrients. Now, if I were to draw a cross section, also the leaf is receiving sunlight, right? And so if I were to draw a cross section of the leaf, right, from the top and the bottom, and see it from the side, and then it would kind of look like this. You've got these cells on the top, and these are the cells that are receiving the sunlight, right? And then just underneath these cells, you've got these long cylindrical or columnar cells. Okay? So the very top cells, they only contain nuclei and they don't have any chloroplasts. And those are transparent cells. That's the upper epidermis of the leaf. Upper epidermis, the tissue that surrounds the top of the leaf. Then this particular layer here, which has got the nuclei and it's got the vacuoles, and it will also have the chloroplasts, a lot of chloroplasts actually. Okay, richest in chloroplasts, so this will be the one that is doing the most photosynthesis and making the most glucose. This layer of cells, this tissue group of cells, is called the palisade mesophyll. Okay, palisade mesophyll, which contains long columnar, lots of chloroplast containing cells called palisade cells. So that is the palisade mesophyll. Now, palisade mesophyll is present right underneath the transparent upper epidermis. So all the sunlight that lands on top of the leaf directly goes into the palisade mesophyll, which contains this rich number of chloroplasts. Now, underneath the palisade mesophyll, we have a air spacious tissue that contains cells that have a lot of air spaces between them, right? Kind of like a sponge. So these cells belong to another tissue called the spongy mesophyll. And the spongy mesophyll cells also have some chloroplasts. So they're also able to do some photosynthesis, but not as much as the palisade mesophyll. So that's called the spongy mesophyll. And adaptation of spongy mesophyll for gas exchange, we already finished. So spongy mesophyll has these air spaces that allow the diffusion of gases. Now, on top of this, we also get the xylem tissue. The xylem vessels are going to come in and bring the water to the leaf, right? So here's a xylem. It kind of looks like an X in a leaf. Inside a leaf, it kind of looks like an X. And then phloem, which carries the amino acids and the glucose away from the leaves, is going to be present on the peripheries of the xylem. Remember, phloem starts with a P and peripheral also starts with a P. So phloem is always present on the peripheries. So central xylem looks like an X or a plus, and then on the peripheries, all of these are phloem. And glucose and amino acids will go into them. So for example, if you're making glucose, right, amino acids are going to go into the phloem, while xylem is bringing in the water and mineral ions to the leaf. Now the underside of the leaf again contains an epidermal tissue, a lower epidermis. And on top of this, it's got these kidney-shaped cells called guard cells. Now guard cells are super important. We've discussed their importance earlier as well. Guard cells help make this airspace between them, and that is called the stomata. Okay, the guard cells, they also have a little bit of chloroplasts for some reason, but they have, most importantly, they're able to control the entry and exit of air particles from inside to outside and outside to inside. So for example, at daytime, when you have photosynthesis occurring, carbon dioxide is going to move into the cells, and oxygen that you're making is going to go out of the leaf. Water leaves the leaf down its concentration gradient, especially at daytime, which generates a suction pull on the xylem, and that is called transpiration. So water also leaves through the stomata. So these are the tissues of the leaf. So let's go back. So again, the palisade mesophyll is a tissue in leaf that is directly exposed to sunlight. So it contains many chloroplasts and therefore performs the most photosynthesis. Spongy mesophyll, this is a tissue in leaf made up of photosynthetic cells, but it also has air spaces, so it allows gases to diffuse around the leaf. Xylem tissue, we've already discussed this before as well. It is made up of hollow xylem cells that don't have any cytoplasm. It transports water and mineral ions from roots to leaves, one-way transport. And it also contains lignin that is deposited in its wall for structural support. Phloem tissue, which is made up of sieve tube elements and companion cells, they were non-hollow cells and alive cells that transport manufactured substances from source to sink, such as from leaves to roots or fruits or stem. Okay, so these are some of the tissues in plants. Now again, guard cells, they form an opening called the stomata through which water and oxygen can diffuse out, while carbon dioxide can diffuse in. Guard cells, they usually open during the day to allow CO2 to come in and O2 to move, and they close during the night because you don't want any CO2 to come in, and if they open during the night, then all the water is going to leave. If water leaves, then the plant is going to be dehydrated, right? And it's going to start wilting. So that is why they usually close at night. Except for some exceptional plants that try to conserve water. Okay. Are you hungry? I could use a light snack. Xylem, we've already finished this, but let's just quickly review this. So xylem tissue is made up of dead hollow cells that lack cytoplasm and organelles. It conducts water and minerals only from roots to the leaves, and so it's a unidirectional flow, and it is lined by a rigid material called lignin, and this is kind of how it looks like. Phloem is a tissue that transports organic nutrients, mainly sugars, and amino acids and lipids from the source, usually the leaves, to the sink, like roots or stems or fruit, and therefore it is a bidirectional flow, because it's not going one way. It is made up of sieve tube elements, so these cells that you see here, these are called sieve tube elements. And each of these cells is alive. How are they alive? Because they have a cytoplasm and they have a companion cell that contains energy from the mitochondria to be able to keep them alive. Okay? And yeah, so this is how the vascular tissue looks inside a stem. We've already done how it looks like in the leaf, right? Remember? In the leaf, it looks kind of like an X in the middle, right? For the xylem, and phloem is present in the peripheries. So you are expected to be aware of how the vascular tissue, the vascular bundle, looks like in the leaf versus the stem. So in the leaf, there's an X for xylem, and there are on the peripheries you find the phloem muscles. While in the stem, what you have is, let's just draw a stem here. For example, you have a stem, right? Here's a stem. And if I were to make a cross-section of this, so cut it here and then represent this, it kind of looks like this. The stem has the vascular tissue that is arranged in the peripheries here, okay? In this place. So this is where the vascular tissue is. Now, xylem is present on the inside, this portion here. So that's xylem. Centrally, again, is the xylem. And then peripherally is the phloem, because it starts with a P. And this is how it looks like in the stem. In leaves, it looks like an X centrally, and phloem is present peripherally. So here's a stem microscopic image, and you can very easily see that we have the vascular bundles here. This is xylem with its wider spaces, and then over here we have the phloems. Okay? Transpiration. What is transpiration? Transpiration is the process by which water evaporates from the stomata of leaves into the atmosphere. So it helps maintain a steady flow of water from the roots to the leaves, facilitating nutrient uptake, cooling the plant, and maintaining turgor pressure. So remember transpiration, right? Transpiration is the movement of water through the stomata outside, which generates a suction pull onto the xylem, which pulls in more water from the roots. So what can affect this transpiration? What can affect the rate of water moving out? Well, one of the things that could affect is the concentration gradient, right? If too much moisture is on the outside, then there's not much of a concentration gradient between inside and outside. So that would mean that there is lesser transpiration. Another thing that could affect the movement of water molecules across the stomata would be kinetic energy of molecules. If you increase the temperature, that would increase the kinetic energy. If you decrease the temperature, then that would decrease the kinetic energy. So increased temperature would also stimulate more transpiration. A third thing that could affect the transpiration rate is the wind, right? If you have a strong wind blowing, so let's say here's a wind, and wind is carrying this water that the leaf very quickly away from the leaf, right? Away from the stomata. Then this water that leaves the stomata does not accumulate on the outside of the stomata, does not accumulate outside of the guard cells, which maintains a steep concentration gradient for the water to keep on leaving, facilitating transpiration. Okay, so let's have a look at your neat notes. So again, factors that affect the rate of transpiration. Temperature, number one. Temperature would cause an increased rate of transpiration because there is increased kinetic energy of molecules. Number two, humidity. Higher humidity reduces the rate of transpiration as the air surrounding the leaves becomes more saturated with water vapor. So this would, higher humidity would decrease the concentration gradient, and lower humidity would facilitate. Wind, again, wind would remove any water that leaves the leaf from accumulating outside of the guard cells, which maintains a steep concentration gradient. And light intensity, of course, because if there is light intensity, then the guard cells will be open. Because during light, you want to do photosynthesis, and for that you have to get carbon dioxide inside, which will stimulate the stomatal opening. So greater light intensity would mean greater rate of transpiration. Okay? Now, a topic on the required practicals, and this is very simple, is trying to find the number of stomata open. So basically what you do is you take a microscope, and I've already taught you how to use a microscope in the cells chapter. To find the mean number of open stomata on a leaf, precisely what you do is you calibrate the microscope, you randomly sample multiple leaf areas, usually from the underside of the leaf, and count the open stomata in each sample area. And then once you have a couple of data, like you get 10, and then you get 11, and you get 12, and then you get 10 again, and so on, then what you do is you calculate a mean, okay? You calculate a mean using all of them. So calculate a mean open stomata per sample, estimate stomata density per square millimeter if you have the area, and then verify accuracy in report findings. Okay? It's very, very simple. And lastly, we have an instrument that you need to be aware of called a potometer. So I'm just going to very quickly tell you how a potometer works. A potometer is something that we use to measure the rate of transpiration. Okay, a rate of transpiration. So how does this work? It kind of looks like this. So you have the plant for which you're going to be measuring the rate of transpiration. So what is transpiration? Remember, transpiration is the movement of water outside of the leaves, right? Water is leaving the leaves. This causes a suction pull to be generated in the roots, pulling more water upwards, right? Now, the faster this water leaves, the faster it will be pulled up from the roots. So as this plant undergoes transpiration, it will pull water from the root, and that is attached to the potometer. Now, the potometer contains water connected to an air bubble here, and we observe the movement of air bubbles per unit time to look at the rate of transpiration. Okay, so if the air bubble, for example, in 5 minutes, right, in 5 minutes it moves maybe 2 centimeters. When the temperature is 20 degrees Celsius, and then we increase the temperature. Maybe we take the temperature to 40 degrees Celsius, and now maybe it moves 5 centimeters, right, to the left. So of course, at 40 degrees Celsius, within 5 minutes, it has moved 5 centimeters to the left. So a lot more water has been transpired, a lot more water has been absorbed by the plant at 40 degrees Celsius, as compared to when we did it at 20 degrees Celsius only, it moved only 2 centimeters to the left, meaning that there was lower rate of transpiration. So this is how a potometer works, and this is how we can use it to be able to compare transpiration rates among different conditions. Okay, so you can change the temperature, you can also change the humidity, and so on. So you can possibly see it on the exam, and usually this would come in a question. So here's a real potometer, and this is how it looks like. Okay, so again, using a potometer works by measuring how much water a plant takes up through its roots and how much it loses through transpiration. It consists of a tube that is filled with water with the plant stem attached, and as the plant takes up water, air bubbles move through the tube, showing the rate of water uptake. So the most important thing is the air bubbles movement. By observing these bubbles and controlling variables like light intensity or humidity or temperature, like we did in our example, we can understand how different factors affect the rate of water loss in plants. Okay? And that's it. So congratulations, you have finished the organization chapter. And yeah. Now make sure that you review these videos a couple of times to instill all the content in your memory. Okay? We're going a little bit fast, because we're trying to cover this in the shortest amount possible, but we are not leaving any details. So I would love that you guys are able to review these videos and review the notes and make your diagrams and make annotations, okay? And again, if you need any help, if you need any guidance or additional tuition, if you need to study any other subjects, just let us know in the WhatsApp link provided below, okay? And I'll see you guys in Chapter 3.